everybody, and welcome to this episode of the Health Span Action Coalition podcast, where we make longevity science accessibly nerdy. So we try to help you understand what's going on. And today, as always, here with my co-host, Dr. Heather Maines. Hi, Heather. How are you?
Hello, I'm doing well, thank you.
And we are joined today by Dr. Lorna Harries. She is a professor of molecular genetics, heavily involved as a founder in a very cool company called Senesca, which we'll talk about today, and focuses her work primarily on senescent cells, which are these quote unquote zombie cells that we hear so much about. But hopefully, we can learn a little bit more today through our conversation about what a senescent cell is, why it's an important therapeutic target for longevity, and maybe not just for longevity, but diseases of aging that we associate with aging at least. But then also, how can we make these better? Brad, like why do they exist, and how can we optimize them? So, Dr. Harries, thank you so much for being here. It's wonderful to have you.
Oh, you're very welcome.
All right, so let's first start off, if it's okay, is what is the senescent cell? Because I think a lot of people that live in this longevity world and biohackers and all these different monikers are aware of cellular senescence as something that is not good. But why do we have these cells in the first place? What is a senescent cell?
Sure. So pretty much all different types of cells can become what we would call senescent. And basically it's just an old creaky cell that's stopped doing the things that it needs to do and started doing a bunch of stuff that you don't want it to do. So pretty much every type of cell in your body has the potential to become senescent, with the exception of germ cells and the cells that we start off as when we're really early embryos. Those cells don't senesce, but pretty much everything else does. Now, I often get asked why on earth would we have bothered to evolve such a thing if it ends up in aging and disease?
Exactly.
And the simple answer to that is it's part of a cellular defense mechanism that cells have evolved to, when things go wrong with them, they can sort of throw, one type of senescent cell can kind of throw a switch to become senescent, to take it out of circulation. The problem is that cell is throwing out a bunch of chemicals to call in the immune system to clear it. Now, as we age, the immune system is also aging. So, you're sending out the signals, but they're not being heard. So these cells accumulate. And when they accumulate, these chemical signals, these inflammatory signals start to accumulate. And it's that we think is responsible for not just aging on a cellular level, but also a whole plethora of diseases that are associated with it.
So as we talk about in these, again, longevity circles, they talk about inflammaging. So is this kind of one of those central responses? So it kind of sounds like to me it sounds like a distress call, right? Like when you're in a boat, say for instance, and the boat stops, you shoot up a signal flare or you send out a distress signal to try and get help. So these are cells that say, you know what, something is not working correctly. It has the knowledge and the wisdom the cell does to know that something isn't ideal. So it sends up these flares. The problem is there's probably so many hundreds, if not thousands of these cells sending off these signals.
Fire to the boat.
It overwhelms our body's capacity to clear them. Am I thinking about that correctly?
Yeah, I mean, that's certainly true of a subset. So the other thing to note about senescence is it isn't one thing. So there's at least 30 different types of senescent cells. Some of them are beneficial, but the thing which unites all of the ones which are not beneficial is that they're unregulated. So, you're throwing up your flare. In fact, as you say, you're throwing up so many flares, and what you're actually doing is setting fire to your boat.
So I mean, as oftentimes we see with aging, these are these normal processes that are programmed in correctly, and then they take an unexpected turn. So that's like what it sounds like is happening with senescence.
That's pretty much true.
30 different types of senescence, which I didn't realize, at least that we know of as of this is 2026 when we're filming. But you said some of these can actually be beneficial to the organism. So what are some examples of beneficial senescence?
So we need senescence during embryonic development. So, for example, when you're developing as an embryo, you actually have webbed fingers, and senescence is one of those processes that removes the webs. It's also really quite important in things like wound healing and tissue remodeling. So, the response of your tissues to external stimuli. But that's a very specific subset of senescent cells. There are other types of senescent cells which are senescent because basically, as you say, they're zombie cells. So one senescent cell can tell all the cells around it to become senescent. And they're a different type of senescence, perhaps, than the initial cell was. Cells can also become senescent because of stress. So unresolved cellular stress, things from both the inside of you, but also from the outside, those also can be a provocation for stress. And you're right. Inflammaging senescence is one of the major contributors to that.
Interesting. So, and Heather, feel free to jump in as we're kind of talking.
I'm
I kind of want to.
because my nerdy upbringing was all about telomeres.
Yes,
and I've worked with embryonic stem cells. So how does this all link into that concept where we were always told in tissue culture, you can't grow the cells for too long or they senesce? Of course, it's not the case for the pluripotent stem cells. So how do we separate the fact that our body cells don't supposed to proliferate too much?
Sure. So the telomere connection, again, this is a type of senescence called replicative senescence. So that's exactly it. It's the cells. And the telomeres get so short that they send signals, basically, which activate senescence. Now, in some cases, there are very good reasons why you may not want those cells to divide. So, for example, if you've got things like activation of oncogenes, that's quite often one provocation for senescence. But in
an
a lot of other cases,
oncogene is a gene that initiates cancer.
It's a cancer gene, basically. Other times, there's actually not much wrong with the actual cell. It's just kind of reached the end of its replicative lifespan. So I like to think about it a little bit like a software problem. So in some cases, your hardware is fine, but you've got a glitch in your software, which means that the cell is no longer functional. Now, what we think, myself and other scientists who are going down this route, is that for those types of cells, if we can actually just reboot the system and reset the software, then we can actually get those cells to behave themselves and stop them doing all the things that they shouldn't be.
So this was my question, actually. Like, is it just evolution trying to knock us off? Because if you make, 100 cells, if you make 100 cells senesce in case something is wrong with them, or, if the cells have a natural lifetime and then evolution's decided, 7 is enough for you. That's enough proliferation. You just shut down. I don't care if you're feeling good. I don't care. Just shut down. So it's almost like risk aversion from an evolutionary perspective
so I think
they kind of want us to die anyway.
I'm not sure I agree with that. This process is actually a really nice example of something called antagonistic pleiotropy, which is a really scary scientific word. But what it actually means is things which are selected for in young life because they're good for you sometimes can have bad consequences in later life. So if you think about it this way, so evolution really is a pressure on reproduction. It's a pressure, passing on your genes, passing on the best characteristics of your genes to the next generation. And frankly, once you've done that, nature does not care what happens to you. You are redundant. So you're selecting for something which is actually good for you while you're still reproducing. And the fact it happens to have so many deleterious, nasty consequences when you're older is irrelevant from an evolutionary point of view. So it selects for the young.
Exactly. Yeah. So then from a Senisca point of view, or from a senescence kind of targeting therapeutics, there seems to be 2 different, or maybe 3 different ways of going. You can either reactivate a senescent cell or clear them. I read a little bit about like autophagy, that you make the immune cells come in and eat them, or kill them, senolytics. Can you go over a little bit, what the different techniques are that we're trying to win?
So it basically falls into 2 camps, actually, I would classify them. So you have the senolytics. That would include, things like bringing in chemicals which would allow the cells to be killed, or you have a senomorphic. So the basic distinguishment between those is with the senolytic, the cells are gone. With a senomorphic, they are reprogrammed, or they are in stasis. So you've kind of reactivated them, but you've stopped them doing what you don't want them to do. So I throw out the chemicals. Now, I think there are pluses and minuses on both sides. So on the senolytic side, the advantage is that the cells are gone. So, they're no longer there causing problems. The issue with the senolytic is that it doesn't distinguish between subtypes. I mean, some of the new 2nd generation ones are starting to do that now. But by and large, some of the 1st generation ones, they hit everything. that includes your beneficial ones. It includes all of the sort of the tissue resident stem cells which do senesce. So you can't actually rebuild, and you're removing a bunch of cells that you can't actually then rebuild the tissue. There's some data that I saw presented at the ICSA meeting this year that also suggests that that sort of large scale apoptosis that you can get from killing lots of cells at once sets up a really nasty pressure in your tissue for the accumulation of more senescent cells down the line. So that's one way of looking at it. The way that we are doing this, and we didn't actually set out to design it to do this. This is just what our point of traction does. It's a reprogramming. So it basically restores the functionality of the senescent cells, but not completely. So it's like a partial reprogramming. So we're able to rejig the morphology back where it should be. They regain the functionality that they should have. They don't, the way we're doing it, we don't actually initiate proliferation. We've managed to uncouple that, so that there's no chance that they can then go and make a clone. We've got some pretty clever biochemistry and molecular genetics that allows us to actually restore the levels of the genes that regulate this within their natural homeostasis, within their natural thing. So these genes normally sit in a Goldilocks zone. You don't want too much and you don't want too little. We found a way of being able to just push them back there, and then the cell will look after it. So it should be, I think, a lot less risky than some of the other kind of reprogramming technologies, which historically have maybe carried something of a cancer risk.
So, I mean, that sounds fascinating to me. So we are repairing the cells just enough that they don't cause problems, right? So that's fascinating. And, it's interesting because clinically, I just think of this as very binary, right? A cell is either functional or non-functional, but kind of it sounds like you're introducing that there's so many different levels kind of
Nothing in biology is ever that clear cut.
You're either dead or not as a clinician, right? Like that's maybe the only binary we have. No. So my question clinically is, when in organisms, in terms of their lifetime, do we start to see senescence become a thing, right? So you mentioned, the webbed fingers and things, right, in embryonic existence. So obviously senescence starts very early in the proper cell lines. So so it's not just the presence of senescent cells, but it sounds like unplanned aggregation of senescent cells becomes the issue. So when do we start to see that, particularly as we talk about health span, right? Are we seeing those aggregate in our 20s and our 30s? Clinically, when we talk about aging, there's two sentinel ages, 44 and 60. For some reason, those seem to be kind of where aging just kind of falls off a cliff.
We know why
right?
Yet.
Yeah,
Thanks,
but those.
Sanjay.
Well, I mean,
I'm getting there very soon.
I am. Don't worry, we're all on the same train here, just different cars, right? But is it that we have these senescent buildups, or that all of a sudden there's some sort of an epigenetic thing that happens after a certain number of cell replication cycles? I'm just trying to think of it because I'm not a smart PhD like you folks, but is there something that just turns on senescence, or is it lifestyle, like live and environmental stress, emotional stress? You mentioned stress at a cellular level, right? So that when we think of cellular stress, that could be reactive oxygen species, that could be, all sorts of things. But, from a clinical standpoint, are there things in our existence that promote cellular senescence?
Yeah, I mean, I think the answer to that is all of the above, actually.
Yes.
So yes,
there's
a burst of senescence that happens during embryogenesis when you're forming, before you're born. It's not we don't yet know whether it's a linear process. I don't think there's a big event that happens in your 40s and another one in your 60s that causes a massive increase in senescence. I think it's a more stochastic, a more random additive thing. The other thing to say is actually in people and in living systems, the actual number of senescent cells is actually typically quite low. So it's not like we start off with none and we end up with all of them. There's likely a threshold, and I guess it will interface with the activity of your immune system. So when you're young, your immune system is coming in there, it's getting rid of them like it should. But as we get older, your immune system becomes less able to do that. So there will come a tipping point, which I suspect is around 50, 55, which is really depressing because I'm 56 next birthday. But if you superimpose that on the demographic of age-related diseases, there's an inflection point at around 50. So actually things like osteoarthritis, dementia, diabetes, cancer, cardiovascular disease, all of these big common chronic diseases, you're pretty okay actually till you hit your 50s, and then things start to exponentially go up. And I think you kind of reach that cellular tipping point, and it might not just be senescence, of course. There's other things that can feed in, but your system is having all of these signals from inside, from outside, some of them regulated, some of them planned, some of them not, which are pushing your cells to do this or to resist having this done to them. And then layer on that whether or not the immune system is actually capable of removing them. So it's a complicated story. I don't think we can actually simplify. None of this is simple.
I mean, I always say the dumbest cell is still 100 million times smarter than the smartest human, right? When you think about all the processes that are happening, and clinically, it speaks to exactly what you're saying. Is as a cardiologist, heart disease happens in your 50s, like diabetes happen, like you have this capacitance up to a certain age, and then you hit a tipping point. Yeah,
I mean,
you have that
the
cellular
definition
resilience.
really is resilience. Cells are beautiful, and I think the more, if you drill down below the level of the cell and you're looking at the mechanics of the genome and how and when your genes are switched on and off. I mean, I know when I came out of my PhD a long time ago, I remember thinking to myself, I've got this, I know this is fine.
Yeah.
And here I am, I'm 35 years later, and I'm beginning to realize that I actually know a titchy tiny bit of it. There's always new stuff to discover, new points at which you can intervene. I think we're reaching a flexion point now, though, that we are beginning to understand enough about the biology of aging and age-related diseases to be able to actually start to impact them now. And I think this is going to be a really, really quite exciting decade in medical research.
I'm very interested now in the immunology aspect of it, that you say that, the immune cells kind of stop finding them and stop getting rid of them in their bad state. If we say that they have like a senolytic bad state versus like a postmitotic just hanging around, not doing any damage. And I did read about effects with senescence in Alzheimer's, Parkinson's disease, and diabetes.
Yes.
Do you see any sort of links here in terms of senescent cells, in terms of immunology, and potentially in terms of like infections? Like when we get hit by an infection, can it hurt our immune system enough that it just stops doing all those little jobs that it was supposed to be doing?
Yeah, so that would count to me. That would count as an internal all external stress. We did some work some time ago, actually, that showed that repeated immune challenge is certainly something which can provoke a stress response in the body, I think. And you put on top of that the fact as we age, your immunity goes down. So your levels of inflammation go up, but your ability to respond to viruses and bacteria goes down. So there's some lovely evidence that's coming out now that if you actually deal with senescence in the system, that you can actually improve things like vaccine response if you kind of give the immune system a little bit of a reboot.
I mean, it almost sounds to me like, on your phone, you have background apps that are refreshing and draining your battery, right? I mean, that's essentially what these senescent cells kind of do to your immune system. So if you're able to get rid of the senescent cells so that now your immune system can focus the bandwidth on the task at hand of viral illness. But I think Heather, so interesting, particularly in this post-COVID world, to think of the burden of what COVID infections have done to aging. And I can tell you so many of my patients that are in that elder age group who had COVID, some of them came out, as you mentioned, Lorna, just resilience. Like, there's one patient that I can think of in particular where he was on a vent and, a ventilator, and I thought for sure he was going to pass away. He didn't. And now he just comes back stronger than ever. I mean, his immune system.
Got good genes.
Yeah, I mean, he does. I mean, he's just a wonderful guy. But, these repeated viral challenges, and maybe as we think about kind of the viruses of society, EBV and, and the echovirus, rhinoviruses, all the things that are around, there may be some degree of like immunological hysteresis, right? Controlled stress that keeps us keeps the immune system charged. But then you get this overwhelming episode, and that just pushes us over a cliff. And as I think about senolytic therapies that are available, one of them that comes to mind in a lot of the different formulations that are commercially out there is quercetin, right, as a senolytic, and that has a very clear immune response. So I think it's interesting that maybe charging the immune system or boosting the immune system to get rid of senescent cells. I'm convinced the more and more there's two things that are involved in longevity. Number one, mitochondrial health, and number two, immune health, right? So this just feeds
they also interface
that cycle.
much.
Oh, for sure.
If we back off this and look at this at the kind of molecular. I'm happier in the molecular cellular arena.
Let's
if you
go. See,
take a
I see, and
step
I tried to
back,
demote you by making you a molecular biology professor, but here you
oh
go.
no, that's
Like,
fine. I'm good with that. But if you take a step back and you think about this through the framework of the hallmarks of aging, so
yeah,
all of these things that you're talking about, the mitochondrial dysfunctions, senescence, epigenetic alterations, and things like that, they actually all interface. So, I mean, I think it's interesting you mentioned the word hormesis there, and that actually fits bang slap with my hypothesis of what's going on here, in that quite often these are things that, a little sniff of something damaging gives your system the ability to prime and to deal with it when you meet it bigger, if you're with me. But it's when when you don't have that resolution and you don't have that sort of recuperation and that resetting and the opportunity to kind of make those changes to your genome and your transcriptome that you need to make, to your RNA and to your DNA, that you need to make in order to be able to respond appropriately next time you meet it. So it's a little bit, in some ways, it's a little bit like a vaccine, right? So, you give your system a sniff of something so you can mount a
yeah.
defense against it. But a lot of these age-related pathways actually are what I would call hormetic.
Yeah, I mean, there's a phrase that we use. You bleed in training so you can survive in war, right? So kind of similar, like
Yeah.
you're getting these low level responses, and so that's why I think it's kind of important to get that annual cold or that flu, as
Yeah.
long as it's not overwhelming, right? So that your immune system is primed correctly. Um, and yeah, I never thought about this may be just a natural
Mm.
like vaccine type physiology that's happening when we get these low grade infections to prime our immunity. So as we see senolytic therapy, one of the things clinically that people will say is, the first human that's going to live to 120 was born today, or like I'm sure you've heard of that kind of
I've heard
bravado.
that.
So but here, rather than thinking about lifespan, right, in kind of with the Health Span Action Coalition, we're more concerned with health span, right? Not to living to 120, but maybe if I'm living to 89 years of age, that day before I die, I'm at the gym exercising and just living my full life. I have my mental faculties and all of these things. So when we talk about the promise of senolytics, right? Assuming there is a promise of senolytics,
Oh,
right?
I think
Which
there is.
are we?
Therapeutics anyway.
Is
Yeah.
it a lifespan? Is it a health span? Is it both? Like what
Mm.
sorts of things are we targeting? And then maybe we can get into specifically with Senesca what it is that you guys do.
So I think like you, I'm actually all about health span. Certainly in the UK, your average person is going to live the last 14 years of their life with at least one common chronic disease. And by
Yeah.
the time you get into your 80s, actually about 10 to 15% of us have at least four. So actually, if we can do something about that, I think that's important. Whether or not you see impact on lifespan, I think you probably will a little bit, because the genes that regulate lifespan are actually quite often some of the genes that regulate health span, because they're so interlinked. But
Hmm.
I don't think that's like you, my sort of drive for doing this has been all about quality of life and about aging well. I personally, I don't want to be in a nursing home by 70 because I'm riddled with osteoarthritis. Like you, I want to be hassling my children into old age and have all of my faculties.
Yeah, no, I think it's so as we look at senolytics in like animal models, like where are we seeing the lift, I guess,
Yeah.
in terms of therapeutics?
Yeah. So in the animal models, and I think you have to be a little bit careful about drawing parallels with animal models. But I think certainly the P16 depleted mouse. So there's a mouse where they basically engineered a kill switch for senescence, so you can basically take all the senescent cells out. That mouse lives about 25% longer. Whether
Wow.
or
So
not
just
that will translate into people, again, this is one of my sort of things about you're trying to infer things from an animal that lives 3 years and apply them to an animal that lives 90 years. So, and we are different,
Yeah.
but
well,
I think there are definitely some insights that you can get from this. And I think you will see some knock on lifespan, but I think the impact on health span and on your chances of getting something nasty as you age are going to be much bigger.
yeah, no, and one of the foundational things in this longevity space is intermittent fasting based on a Canada study, right? So looking at Candida that were starved, and I think it was an exactly 25% longer lifespan that they had. And so everyone's like, oh, well, if it's good enough for a fungus, it's good enough for us. But we are not just more complicated fungi, right? So many of the processes
We
and
don't
things
quite know what
involved.
fungi are, actually. I mean, they're something completely different to anything else we've seen. But I mean, the calorific restriction, you do see that in rodents, you do see that in other mammals. I think there's some data coming out in non-human primates now. And there is a body of evidence actually in people that you are seeing some restoration of some pretty key health pathways from intermittent fasting.
Yeah. No, but it's just interesting, like, so animals are not just little humans, right? And
No.
I think the point and interesting, I mean, the IF data, the intermittent fasting data is interesting, but the point is that you can't necessarily relate animal studies to the biology of humans, right? And
No.
so it's so much more complex, but at least it's a thought-provoking experiment, right? Like
Of
it's like,
course.
oh,
And
I wonder if that happens in us too.
of course, and there are things that, I mean, for complete transparency, my academic labs are completely animal free, because I think the best test organism for human disease is humans. But that does obviously impose limitations. We do a lot of population work, a lot of sort of molecular epidemiology, that sort of thing. But I think you've got to look at these things holistically. And I think no system is ever going to give you a perfect answer. And I think you have to use all of the tools at your disposal to be able to unpick the biology.
So what are
Can
some of
I
those? Oh, go
drag
ahead, Sally.
back
Go
to
ahead.
if I drag back to the sort of function, the mechanism of action. So you said that we have xenomorphics and we have senolytics. The
Yeah.
xenomorphics are attempting to kind of reactivate to the point of getting rid of the bad senescence signals. So it's not reactivating to proliferation and cancer formation. The senolytics are looking to clear out the cells. So if we come back to, Alzheimer's, Parkinson's disease, diabetes, what in the world is the mechanism of action of how you would get these diseases in regard to senescence, and how
Oh, yeah.
potentially is xenomorphics or senolytics
Sure.
against that?
Okay, so all three of those diseases are very well known to have a senescence component. Diabetes, you've got, if it's not well controlled, you've got a pretty unrelenting metabolic and or inflammatory stress. That's going to induce senescence. And the question of the brain disorders, the brain is an incredibly energy hungry organ. So, you've got a lot of reactive oxygen and things like that, which are going to set up a stress. So, I mean, I think that's pretty cut and dried that senescence is important. And there are also data, both from animal models, but also in people, that senescence is linked. So some of the work that we've done actually a long time ago, we did with Luigi Ferrucci, where we were using at his Inchianti study, which is a human population. It's a cross-sectional population, so it's just a bunch of people usually healthy, but there will be some disease folks in there also. And they took samples from them and they followed them for a bunch of years so that you can actually see what happens to them. And there are people in there who do go on to end up with things like Alzheimer's. And what we found, that the systems that we are working on, which underpin, which are a driver for senescence, those are actually predictive of cognitive decline. So when we looked at people at baseline, these are healthy people, no signs of any brain issues at all. We took blood samples, we measured all of the RNA messages that these people had, and we looked specifically at the messages for the genes that we like. And what we see is the people that have the lowest levels of those messages are the people who go on to have the biggest drops in cognitive function 12 years later. And not just that, they're also the people that go on to have the biggest drops in things like grip strength, which is obviously a marker of systemic frailty. So senescence, I think, it's a really important driver of multi-system disease.
So if we put that in order, then you're saying that the senescence comes first.
Yes.
And then the immune system is just doing what it's supposed to do. Because I typically saw it as like autoimmune disease, that for some reason, senescence, the immune system starts attacking islet cells or attacking dopaminergic
No, I mean,
neurons.
that's a bit different. So that's what you'd see in type 1. So type 1 is autoimmune. But again, it's in type 1, you have heightened inflammation, and inflammation is a really big provocation for senescence. So my view is that senescence is causal of a lot of these diseases, but it is also a consequence of them. So it's a little bit like a vicious circle. So, let's take diabetes as an example. So you start to accumulate senescent cells so that you start to lose the functionality of the organ. You start to be able not to regulate your blood sugar properly. The fact that your blood sugar is fluctuating wildly means you've got an ongoing stress, which is going to set up more senescence, which is going to impact your organs. So it's I think that's one of the reasons why this shows so much promise as a therapeutic approach, because if we can break that cycle, then we can maybe do something about it.
And as you speak specifically about diabetes, or as we look at Parkinson's or Alzheimer's, is there a possibility of reversal of disease? I mean, so, I mean, with central nervous system issues, because the generation time of those cells is so long, I don't know
Yeah,
how quickly they would recover. But when you think about pancreatic cells, or
yeah.
let's say renal disease or
Mm,
liver disease, or, like heart tends to not, cardiac myocytes don't double that quickly, right? But when you look
yeah.
at these tissues that are more rapidly reproducing, is there a possibility to target, for instance, diabetes and reverse diabetes
Mm.
just through senolytics alone, or
So
in
I
combination with other therapies?
yeah, I think it is. So again, this is a really good example of a different type of senescence. So you're right that not all cells have the same division kinetics. So some of the really interesting cells don't divide. So brain cells don't divide. The cells which some of the cells with the biggest set of connections in your body. You've got nerve fibers that go from your toes up to your brain. Those cells don't grow. Pancreatic beta cells, after they have a certain capacity for growth early on in the course of disease, you do see a little bit of expansion in response to things like too much glucose or too much fat in your blood. But then you get to a point where they can't. And typically, the more complicated a cell is, the less division potential it has. But that doesn't mean they don't senesce, because
Hmm.
they maybe wouldn't senesce through the attrition of telomeres or through your telomeres getting shorter, but there are other pathways to senescence, and stress is one of those, as I said. So in those tissues, it's probably stress-induced senescence actually, which is more than replicative senescence, which is important. But I think provided the way that you're intervening to either remove or reprogram those cells is able to deal with that, then yes, I think there's a potential you should see some benefit.
Wow. I mean, it opens up a whole new approach to
Mm.
this, right? And we haven't talked much about stem cells, I guess, in this. And I don't know if you have any background, but you know,
Not a sausage.
not a sausage. I love it. But when we talk about cells that have ultimate potential to not be senescent, right?
Mhm.
Like, and maybe take on the characteristics of the local host, that may be part of the benefit of the stem cells. So if we don't have a sausage, we're not going to kind of follow that thread.
No,
But
no, I
but
mean, actually, I
but
think stem cells are super interesting from that perspective, because there is something about them that allows them to maintain that kind of, proliferative, rebuilding potential that you don't have in other cells. And I think that if we can identify what that is, that might be another really nice handle to actually do this.
and
And you
as
also
we talk
did
about,
say,
sorry, go ahead.
sorry, you did say before as well that some of the earlier senolytics technologies actually wiped out those senescent stem cells.
Well, the tissue resident ones, yes. So
Yeah,
obviously, I'm not a stem cell biologist, but I know you start off with a stem cell that can basically be anything. Those ones,
pluripotent stem cells. That's my
exactly.
field.
Those ones don't senesce. Once you start down that differentiation pathway, so what you'll find is that there's a population of cells, for example, in your heart, which they're not pluripotent, they're not capable of being anything, but they can rebuild heart cells. Now, those ones, they've gone a little way away from where they started, and somewhere between the ones that can be anything and the ones that have a restricted bunch of things they can be, they have acquired the ability to senesce. That's, I think,
And
a
this is
really
always
interesting.
So
this is something
so
very much mesenchymal stem cell field, because when they're trying to do expansion of mesenchymal stem cells, they
Yeah,
always hit this senescence wall.
exactly that.
And
Yeah,
so they can never scale mesenchymal stem cell
exactly.
expansion.
So this is one answer
They
to
have
that. So
worked out this senescence wall.
yeah, so that's actually one really, really nice application of this technology, is in things like sort of mesenchymal stem cell therapies, external stem cell therapies, CAR T therapies, things like that, where you can't keep these cells growing ad nauseum because they'll just grind to a halt. This will be a way that you can actually, up the yield so you can get therapeutically relevant amounts of them. And there's
immortalized without
not immortal. They're not.
I mean, that's. I almost think of it like suspended animation. Do you remember? So you kind of have them ready to pounce, but they're in that like coiled spring phase where you get them needed. And I think that this concept of tissue resident stem cells is something that a lot of people don't realize, because if we didn't have these, if you had a cut, right, it would never heal. Right. So we all have stem cells available to us. And I think, I was recently in Hawaii and we're out snorkeling and I got some reef rash, we'll call it, like, and so I have these abrasions on my feet. And as I look at them, that was 2 weeks ago, it's still there. Whereas my daughters, right, who are 16, like they're healed. Right. And I'm like, oh my God, my stem cells suck. Like I need to do something kind of more here.
Interesting, actually. So that's the one thing that they did see in those mice where they remove the senescent cells, is down the line some of those mice are getting issues with the ability to remodel their tissue. So the example you've given us there of sort of healing of your skin, that's a remodeling. They found that in the lung, some of those mice that had all the senescent cells taken out were now having problems remodeling and repairing their lungs. So it's a balance.
As anything in life, I think you mentioned that Goldilocks, too hot, too cold. We want to be just right.
Goldilocks.
Yeah, it's amazing.
And now that we've hit on my stem cells, I want to return to something that both of you said, and I was like, I have no idea what you're talking about. You said hormetic. What in the world is that?
So it's hormesis. It's the concept that a little bit of something can be beneficial, even when a lot of something isn't.
Right. Okay. Like arsenic.
Yeah, I guess so. Yeah. Although that's not what I'd be volunteering for. Yeah.
Back in the day.
But
Okay,
it's like,
sorry, that was
like controlled, controlled stress.
Yeah, it's
a way and coming back on my
primed.
Being on my pluripotent things again. Like we typically say, because we like to market our field, that pluripotent stem cells are the only naturally immortal cell type that is not cancer.
Yes.
So when I was meaning in terms of could you regulate that senescence to make the cells, let's say, temporarily immortal, that you could keep generating more copy for the sake of scale expansion, and then take it away and let them.
And they stop exactly that. So we've shown this actually in my lab, that we can, the cells, once you've reprogrammed them, they're not immortal. They will still senesce, but they just senesce slower. Yeah. So you can either take young cells and slow it down, or we have actually shown, and what we're doing in the sort of the corporate side of things with the drug development we're doing, is to actually show that you can reprogram and reverse senescence.
So let's talk about the drug development that you guys are doing, right? So where do we start, right? Because you mentioned there's 30 flavors at least of senescence, right? And you have the ability to, I guess, mute some of these senescent kind of the deleterious interleukins and cytokines and things that are launched by these. So where is the first therapeutic application? Is it like I think in the space, oftentimes health span, lifespan gets confused with anti-aging, right? And so like, as a skin thing, as a cellular thing, as a Alzheimer's thing, where are some of the earliest
so we're actually doing that right now.
Targets?
So we started off, so our the way we're looking at things is we actually have a sort of hybrid model that we started off, where we have actually gone for skin, because people get very sniffy about skin, that it's it's not all that important, it's all about how beautiful you are. Actually, it's really not true. Skin is a really, really important organ, and it impacts on the health of the whole system. So we are developing some new skincare based on our concept. And we're using
based on oligos.
No, that's not the oligos, that's molecules. So we have molecules and oligos. The molecules hit the top of the pathway, the oligos hit the bottom. So with the skin, we're using, obviously we wouldn't want to use an oligonucleotide in that context, because it would need obviously regulation.
Well, I was going to say you probably need a prescription and physician involvement and all sorts of things. Yeah.
So we are using combinations of natural molecules to hit the pathway at the top. Obviously all the safety talks and everything is being done to make sure that they're safe, and the molecules we're putting in the top end are known to be safe. But we can show real traction, actually, not just on senescence but on proliferation. So we don't see proliferation. We're also seeing repair. So the DNA damage is going down. We are seeing drops of inflammation. And then on the other side of things, so that's the skin. Where we are actually looking at from a drug development perspective is at IPF. So this is a condition called idiopathic pulmonary fibrosis. It's a condition of fibrosis of your lungs. So your lungs basically lose the ability to expand. It's got a really, really awful prognosis. So folks, after diagnosis, they rarely see 3 years after diagnosis before they're dead, because there's just no cure. There are some treatments out there and a new one approved this year, but they're palliative, really. They're not disease modifying. They just slow things, and not for everybody. And quite often they have really horrible side effects. So actually doctors don't want to put their patients on these because their quality of life is so terrible. What we're doing is we're doing something a bit different, that we are harnessing a pathway that we discovered that actually is intricately linked with senescence. And when we do that, we are able to, in end stage lungs from patients with IPF, so we look at them ex vivo, out of the body, we are able to not just take down senescent cells, but also, again, we see the improvement in DNA repair. So the cells are repaired. We don't see proliferation, but what we do see is resolution of fibrotic markers. So we see a repair of the fibrosis in end stage lungs from patients. So, we're not just looking at slowing this down, we're actually looking at stopping it, if not reversing it.
Well, and that's fascinating because as I think about, I think of TGF beta as the fibrosis kind of thing, right? So when you look at, how many diseases associated with aging have fibrosis as their issue, right? Certainly heart, right? Like post-myocardial infarction, what's called
Kidneys, heart, livers,
diastolic dysfunction, heart, liver, pancreas,
everything.
renal. I mean, if
Even hair
like,
loss. Hair loss is due to fibrosis in the follicles.
and then all those people going to Turkey who don't need to go to Turkey anymore for their hair transplants. But it's fascinating, and as I learn, I'm not a PhD, right? But on the MD side, as I learn about this and you understand the underpinnings of aging, fibrosis is so central. And I think starting out with IPF is smart because there is nothing else in that space that's really
and
doing
it's known
anything.
to be driven by senescence. So
Yeah.
it's there's a reason why a lot of us who are working in this area go for IPF, and it's because it's one disease where it's unequivocal that senescence is the driver. There are genetics that tell you that. But what I will say is that the processes and the pathways that we are targeting, that we discovered, are so ubiquitous that we've seen traction in lung, in skin, in joints, in brain cells. It works everywhere. So really, the world's our oyster. The challenge has been you've got to start somewhere. So IPF was where we started.
So that can be my last question then. On these ones where you're looking at disease, are you going for the oligonucleotide solution, or is the oligonucleotide?
Yes, this is oligo, because this allows us to be very precise.
So what is oligonucleotide? Sorry.
Right. So oligos are
simple cardiologist. I don't know these things.
Little bits of DNA or RNA that stick to the messages from your genes and do stuff to them. So some of them usually will chew them up. We are doing something a little bit different, that we're blocking something that would otherwise chew it up. So we're actually switching things up rather than down. But they are the next frontier of medicine.
So I understand them as small DNA sequences.
They can be RNA, they can be DNA. We're using modified RNAs.
So how do you deliver them?
We don't. You see, this is the beauty. So because this was another reason for going for the lung, we are delivering them by inhalation, and we do not need any nano carriers. So when you have your COVID vaccine, for example, it's in a little bubble. We don't need that. This is just in saline, and we can just breathe it in, and it goes in.
So you're just aerosolizing.
When you put it into the blood, it goes over the whole body, right?
Yeah. So no. So it's local delivery straight to the lung.
Local first, and then
yeah. So that's the big elephant in the room for oligotherapeutics. That delivery is a challenge. You can get them into the liver really nicely, and you can get them into the lung through inhalation, but getting them anywhere else is and remains a challenge. I mean, there are people working on it. There are people who've managed to successfully get them into the brain by sticking a molecule onto them to postcode them, if you like. But this is an evolving science.
Yeah. And so just even stepping back, so I think a lot of people are very aware of peptide therapy. So peptides are the proteins, but we're talking about going back a step or two into the DNA and the RNA that codes for these proteins, delivery becomes an interesting issue. Like, and so a lot of times most people think of like viruses, right, to be able to deliver these sorts of therapies. But I mean, it's fascinating. My daughters are taking biology right now, and I explain these things to them. It's fascinating to me what it is that we get to do, right, and where these therapeutics can go. I mean, you're delivering bits and pieces of DNA and RNA into cells, right, locally. And I think the lung is a great, because the inhalation shouldn't really have a lot of systemic things, but just
it
absolutely.
doesn't. We've measured that. So we've looked, obviously we're having to use animal models for our safety and for the to look that things get where they should be. And what we have seen is by a respiratory route, we see it in the lung. We do not see it anywhere else.
Fascinating. And because I think also we have to realize that naked DNA in the stream can be an immune kind of target, right? Like you're not supposed to have that in there, or like mitochondrial DNA, you're not supposed to have that. But
so these are RNA for a start, and it's modified. So it's not a type of RNA that you actually find in nature. So it's not recognized as an immune provocation.
You're so smart, Lorna.
I don't know how good that
like that is.
I've been doing this a long time, Sanjay. So.
I mean, this is so just cool, right? I mean, like, as we were talking about making nerdiness accessible, I mean, this is fascinating to think of just how long ago did we discover DNA, right? In the course of human history. I mean, not that it was discovered, but kind of, like realize that was the method. Watson, Crick, and Rosalind Franklin, right? Like
I think it was 1953.
Was it 53? That sounds about right.
Don't quote me on that.
Well, somebody will fact check us on, the internet loves to fact check us, right? But I mean, to go from understanding that this was the molecule of human life to now bending it to our will to improve immune response and cellular health, like, wow. I mean, just absolutely fascinating. And I wanted to go back. I wanted to go back because you mentioned something about how you don't have animal testing in your lab, which is, a great choice. As one of the problems with any of these human-based longevity therapies or health span therapies is we have to rely on surrogate markers, right? So because the generation time of humans is much longer than the generation time of fungus, right? And clinically, there are so many tests available for human longevity, right? So telomere age was one, right? There are glycan tests and epigenetic tests and things. So I'd love to get as someone who does on the research side of this, what are some of the things that you look for? Because telomere length is interesting, because I can meditate for 2 weeks and my telomeres will get longer, right? So how much, like, how important is that for long-term ability to kind of assess aging, right? It's like you want something that is changing, and there's a lot of movement towards pace of aging clocks as opposed to how old are you, right? So not necessarily, you are, I'm chronologically 49, but biologically 83. That may not help, but it may be helpful for us to say, oh, for every one biological year, you're aging 2 genetic years, or whatever it may be. So as you're looking in your lab, again, getting back to the clinical biohacker space, at these markers that you use in your labs, just what are some of the things that you use to estimate age?
So I think that the tricky, I mean, this is really the holy grail of the field, right? So something that you can measure in the circulation that will give you a definitive answer.
That's why I asked.
We're still working on it, is the answer to that. I mean, telomeres are really interesting. So the thing with telomeres is A, they're really hard to measure, and B, that they are really variable. So within the same population of cells in a 10-year-old, you will have an overlap of telomere length with a 60-year-old. So the longest telomeres in a 60-year-old will overlap with the shortest telomeres in a 10-year-old. So this is a distribution, and it's not as simple as it's this therefore equals that. The same thing with the epigenetic clocks. I mean, I think they're really, really good at telling you how old you, how many times you've been around the sun. But I think you have to ask, what actually are you measuring? So are you actually measuring age, or are you measuring a surrogate marker for age, which you almost certainly are? And how does that impact on the function of your body? I think the new clocks are starting to incorporate those functional measures in. I don't think there is one marker that you can use. I think you have to craft your markers according to what you're doing. So, for example, in the work that we are doing in the lung, we are looking at a marker called MMP7, which is a marker of bad remodeling in your lung. And that isn't an aging marker as such, but it's a marker of the functional impact of aging in the lung. And it's one that people are using in the clinic to evaluate drugs. So that's really the big issue that we have, and that lots of us are thinking about, is how to bridge that gap between what's a marker of aging related processes and what's a marker of disease, and where is the middle ground there between them. There's consortiums being set up. I'm part of a couple of them to be able to establish this. But I think what you're looking at is a composite of measures. You're not looking at one thing which will tell you this. You're looking at, you're taking data from a bunch of sources. And on the kind of more micro level, this is what we do with senescence, right? So there is no one marker of senescence. So when we are determining if a cell is senescent or not, I'm looking at its size, its shape, how big its nucleus is. I'm looking at whether or not it's got lots of lysozymes. I'm looking at whether or not you've got stress fibers in it. When cells start to get old, you get these stress fibers. I'm looking at a whole bunch of things to determine whether or not that cell is senescent. And I think you kind of build that up onto a systemic level, and that's what we're looking at as for aging markers. There's not going to be one. There's going to be a bunch of them that you need to kind of put together.
Yeah, no, very cool, very interesting. I mean, this whole field is just fascinating, right? I mean, obviously we're nerding out about this, but like just so cool.
I was going to say, I think we should do this because it came up in our last podcast right at the end in terms of, senescence. And we were like, ooh, we should bring in senescence next. What would you say we should look at next, Lorna, in terms of aging regulation?
It's almost like a passing of the baton. I love this.
Oh my goodness.
Yeah.
So I mean, I think one of the things that I am really super interested in, and I wish I'd got time to look at it, is the role of the microbiome.
Right. Oh, we have a contact, right, Sanjay?
We've got so many microbiome people.
So we know, we know that if you transplant the gut microbiome of a mouse with diabetes into a mouse without diabetes, that mouse will then develop diabetes. Same with obesity. You can put the gut microbiome of an obese mouse into a non-obese mouse, and that mouse will get fat, and vice versa. So I think that, you've got more bugs, cells from bugs in you than you have cells that are you. And they have such a really, really big impact on all aspects of your biology. So if I was going to, obviously I think senescence is the thing that everyone should be doing, but if I was going to suggest something else to play with, that would be it.
I am much too immature for this topic, however.
It's fascinating. No, it brings to question, who's truly the puppet and who's the puppet master, right? When it comes to microbiome,
exactly.
Because a lot of what I talk about with patients and is really optimizing the microbiome. For instance, GLP-1, right, is all over the map, but your microbiome controls your GLP-1 secretion. So if you improve your microbiomal health in the sense of you get greater microbiome diversity and feed the right fibers, you can just naturally boost GLP-1. There's nothing magic about it; it just happens,
actually,
right?
GLP-1, in my opinion, is the first gero drug. If you look at what it's doing, its benefits for a whole bunch of diseases, you have more benefit than you can account for by weight loss. So I think it's one that's actually one of the very first generations of multi-system, multifactorial drugs that work on actually the fundamentals of aging.
Oh man, we're gonna have a fight now, Lorna, because I think that the majority of it is actually resolution of visceral fat. I think visceral fat unwinds us all, and you have all these adipokines and things.
Undoubtedly, that's really, really important. So people have looked at the benefit that you would expect purely from resolution of fat, and the benefits you're seeing outweigh that.
Yeah, I mean, I've had so many patients, for instance, with like autoimmunity, right? They'll have like joint pain, and they microdose on a GLP-1, and within one dose or two doses, they feel better, right? So, that might be another topic. Maybe not for this podcast, but it's just it's fascinating, right? But as we go into this root cause kind of approach.
The GLP-1s though is as soon as you stop taking them, all the good stuff stops. That's the problem.
Yeah, no, and that's why we have to associate diet, lifestyle, and kind of you can't just you have to fix everything else around there, right? So it's a fascinating discussion. Thank you, Dr. Harries, for being here with us. So just I guess last question. 10 years from now, right? You have a time portal, and you can see 10 years into the future when it comes to senescence. right? Is this something that I can go to my local drugstore and pick up over the counter? Is this something that my longevity medicine physician is prescribing for me? And who gets it? Is it all of our patients with diabetes and Alzheimer's? Do you get it just prophylactically after the age of 50? You mentioned 50 is when stuff happens. Like, what, in the most perfect of worlds, as a scientist, not as a CEO founder, but as a
Yeah.
scientist, like, right? Like, what does this turn into for us in
Yeah.
2036, I guess, is 10 years from today?
So I honestly think this is going to be game changing in the medical field. I really do. And for what it's worth, you can already go down to your local whole food health shop and get chemicals that will do this. So you will have heard the buzz about Ficitin and quercetin you mentioned. You know, they might not be very efficient at it, but they are capable of doing it. I think, casting my eye 10 years in the future, I think this will be routine. I think this will be a new class of disease modifying treatments that target the causes of the disease, not just the consequences.
I think that's great. So, oh, Heather, any last thoughts?
No, this was fantastic. It was so much fun. Thank you for coming.
Oh, you're very welcome. I really enjoyed it.
Absolutely. I love nerding out with fellow nerds. It's so great, right? That's the theme of today, is just like you said, keeping nerdy successful. So own it, love it, and realize how much of the scope of human history we can change, right? By these fascinating things that we're allowed to do. So again, Dr. Harries, thank you so much for being here. And to our listeners as well, thank you guys for being here. It's so fascinating to have these discussions. Hopefully you learned something. I certainly learned a couple of things today. Number one, there's not just one type of senescent cell, there's many. And number two, that senescence starts at birth. All right. Otherwise, we'd all be able to have web fingers and be much better swimmers. It's funny. I was just out surfing the other day and somebody had web gloves that they were pushing out with. And I'm like, oh, that seems so smart, right? So if we kept those, if we didn't have senescence, we'd all be much better surfers. But there's that. But then also learning about how down the road, really harnessing the power of senescence and anti-senescence, if that's a term, can lead to therapeutic resolution of so many diseases. So thank you. Fascinating talk. I guess we're focusing on microbiome in one of our future lectures or one of our future episodes, which just makes a lot of sense. So if you guys enjoyed what you heard today, thank you. Wherever on your screen the like button is and the subscribe buttons are, make sure to like and subscribe, leave comments so we can listen back and take your feedback. And we'd love to hear from you guys. So we'll catch you on the next episode of the Health Span Action Coalition podcast. Bye, everyone.